morning, everyone. Welcome to your last class of 3301. Hopefully, hopefully your semester is going or has gone well, both in this class and in all of your others. And you can kind of take a little bit of a victory lap this week. Come in and do your last class for all of your different things before you get started on studying. I think looking back to my own memories of this time when I was an undergrad, I actually quite liked this period. It was really nice having classes be over. The lifestyle of studying full-time for exams is actually in some ways a little bit easier not easier exactly, but more pleasant than going to class back in the days when you had to physically go to class and wake up in the morning and do things in person on time. It's always nice to just sort of set a schedule and devote, several hours a day for each class or whatever it is that you need to do. And of course, you have the lure of the Christmas holiday coming up after that to motivate you. So lots of stuff to look forward to, and a whole year's worth of accomplishments under your belt. So that's good news for everybody. I guess I don't really have a whole lot more to say by way of introduction. Let's just jump into the content, and then I'll get out of your hair. So today we'll be talking about societal impacts and other considerations. And this is a little bit of I guess you could say a bit of a dessert lecture after eating your vegetables for the whole year, learning about all kinds of complicated topics. I thought maybe it would be nice to spend the last day of class just surveying some of the softer sides of this topic, some of the more morally and culturally relevant sides of genetics and epigenetics, because these things do have important real-world implications that we have to consider. And now that you are all, to varying degrees, knowledgeable in this topic, much more so than you would have been when you started, and much more so than the average person on the street, you do unfortunately acquire some level of responsibility, I suppose, to try to make the best use of this information that you can. This includes being able to, size up findings that you see printed in the news media or in the scientific literature, determine what those findings imply, whether they can be relied upon, what it means for the bigger questions in the genetics of mental health, these kinds of things. And at the same time, if you have any interest in pursuing these topics yourself later on as you go forward in your careers, I think it's worthwhile knowing about some of the challenges that the field still faces. So we can talk about this today, and we won't have to see any heat maps or any Manhattan plots or any of that kind of stuff, and hopefully that'll be pleasing to your minds. But before that, we do have to discuss the unfortunate matter of the final exam, which we do have to have. This is required. 40% or so of your grade still is on the table. That's how much the final exam is worth. And maybe it's 30%. I can't remember what the actual percentage is. No, it's 30%. Sorry, 30%. So 30% of the grade in this course you still have yet to earn, and we have to come up with some way of giving you a number for how well you did. There's probably a lot of ways to do it, but the final exam is the traditional form, and I'm running the final exam in this course the way I have for the other final exams I've done during my time as a contract instructor here at Carleton in the pandemic years. So the final exam is open from December 11th at basically midnight December 11th. All the way until the minute before midnight, the very end of the day on December 23rd, which is the day the last day the university is even open. So you have quite a long time to do it. It's available on Brightspace anytime during this window. So as similar to the midterms, you can log in and take the test whenever you feel like you're ready to take it. I've used this format in the past with 1201 and with my special topics class, and it seems to work reasonably well. I don't know if who knows how it'll go for a class of this size and with a topic of this complexity, But it seems like you don't even really have the ability to schedule an exam if you host it on Brightspace this way. I don't know. The whole exam thing is very messed up and confusing for me as a contract instructor. But anyway, this seems to be a perfectly reasonable way of doing it. So you can choose your own time to do it and spend as much or as little time as you'd like studying before writing the exam. So obviously you would have to consider if you're planning on traveling at some point, you would probably want to get the exam done before you travel so you can just wash your hands of it and walk away. Nothing's better than finishing your exams and being done. But you have to balance that with your need to study and manage the other evaluations and exams you're dealing with. So won't comment on your own time management skills, but that's those are the stakes, and there's no way that I can possibly offer you a extension or anything like that on the exam. That's the domain of the registrar's office. So unlike with the midterms, if you have some problem with your ability to write the exam during this time, that is something that I can't help you with. You'll have to contact the registrar's office, and then they will arrange a deferral or whatever it is you might happen to need. The format of this exam will be similar to the previous ones. I think I've settled on a format that works fairly well. So as far as number of questions, there's going to be about a 50-50 mix of machine-graded questions, meaning multiple choice or fill in the blanks, that kind of thing, and written responses, meaning short answers. The short answers have the same sort of style that you're used to. They have a similar focus as the previous exams, and I'm trying to say there that hopefully after having written several exams in this course, you sort of realize the way that I choose my questions, and you can look at the topics we've discussed since the last midterm and guess what sorts of things I might ask about and what things you should focus your studies on. Focusing on big picture material, of course, is always preferable. Topics that span multiple slides within the same lecture, things that take multiple slides to explain, or things that span multiple lectures, things that keep coming up again and again. The duration will be 2 hours, and you'll have about 30 questions. Although, I don't know, maybe actually less, because normally I give you 90 minutes, I think, and that's usually around 20 questions. And lots of people have problems getting the whole thing done in the allotted time. So yeah, probably 30 is actually too much for 2 hours. Anyway, maybe more like 25. We'll see. I haven't written all the questions yet, so I'm also kind of giving myself leeway to write fewer questions because it's a fair bit of work to come up with these questions. Anyway, but it'll be let's say this. It'll be a density of questions and time that is equivalent to the previous exams, give or take. I certainly won't try to make it any longer, and I don't think there's any benefit to having you all run out of time before you finish all the questions. I think that's a big problem. So we'll try to work on the question to time ratio and see. Like I say, the exam's not made yet, so I can't tell you for sure. The coverage, I'm planning on having it be about 50% material post midterm, meaning lectures that begin with obesity and continue all the way to today's lecture. And then the other half will be sort of cumulative questions that ask you to integrate concepts that span multiple lectures across the entire course. So there, I'd like you to reflect on topic areas that are sort of recurring themes in the lectures that I've given. As a starting point for hints, you can think about questions of cell composition. I've often brought this up as a major confound in studies of gene expression and epigenetics. Linkage is a common topic, how it is that linkage structure in the genome enables you to locate disease-associated genomic regions. Also, how it enables you to impute missing genomic data from genotyping arrays using linkage disequilibrium. You can impute SNPs that you don't have data for, things like that. There is repeat expansion. This is something that is responsible for a variety of conditions, and it's also used in the context of, for example, ASD as a way of as sort of another genetic basis of ASD. There's questions that I could ask perhaps about gene ontology or annotation, the way that you can take sets of genes, whether they're from GWAS studies or RNA sequencing studies or epigenetic studies, and ask what categories or functional processes those genes all relate to. This, you kind of, maybe you've noticed I've had a theme I haven't had the theme, but the research itself shows that many conditions have a neurodevelopmental origin or have an origin in the sort of developmental processes that shape the brain. So that information comes from analyses involving like gene ontology analyses and things along those lines. So you might start to think about that, like how you could unite this common theme where all of the genes linked to like a variety of diseases, from obesity to ASD to depression and more, all seem to involve neurodevelopment in some way. And you might think about how I could ask you questions that ask you to integrate all that kind of stuff. We could also talk, for example, about the success or failure of candidate gene approaches. I've brought that up a fair number of times. Often there's a lot of candidate gene literature, whether it's in depression or in schizophrenia or whatever other topic, where people have in their mind a gene that they think is important for the condition, and they'll do all kinds of studies where they only look at that one exact gene or one exact variant inside of that gene. And those studies, as we've seen, tend to have a lot more, they tend to have on the individual level a lot of successes, but on the cumulative level, when you look at them all together, they don't tend to work terribly well. So I might ask you a question where you'd have to kind of explain that idea. Genetic correlation comes up a lot. I've hinted that the fact that so many of these phenotypes seem to have a genetic correlation might imply that there's an underlying causal architecture that transcends the diagnostic boundaries we've established using the DSM and other kinds of psychological tools. The fact, for example, that depression, schizophrenia, and bipolar disorder are all genetically correlated, meaning that SNPs that increase your likelihood of, say, schizophrenia also increase your likelihood of bipolar disorder and depression. The fact that we see this does seem to imply that there's maybe like a common mechanism or some kind of common process that's disrupted in the brain to varying degrees. And the difference between these conditions may not be one of kind, but it might be sort of a more quantitative sort of difference. I mean, it's hard to really say. But I'd like you to reflect on that. That's a pretty important big picture topic that you might be asked about. And then DNA methylation, we have a lot to say about that, of course, not only because it's my own personal area of expertise, but also because we have conditions like Rett syndrome that rely heavily on DNA methylation, as well as things like developmental programming, the concept of epigenetics more broadly, and so on and so forth. So those are just some hints as to bigger themes that I might ask you about in the in the exam. Most especially in this half that's cumulative, that asks you to integrate information from all the different lectures. So that's all I have to really say about that now. Hopefully that's given you a good starting point for your studies. I guess, it's funny to be in a position where you can theoretically go back and rewatch all of the lectures. This was not available to students prior to the advent of online learning. I don't know. I mean, you'd have to think it's helpful, but I guess you kind of have this trade-off where if you know that you're only going to see a lecture once live, you'll be much more careful about taking notes from it. And that act of taking notes itself is really quite essential for learning. You really have to pass the information through your ear, through your brain, through your muscles, and onto paper or onto a tablet or whatever it is you use to take notes. That really does help quite a lot for learning. And that maybe is not incentivized by the fact that you can just listen to the lecture whenever you want, and therefore notes are sort of redundant, especially also since you have the PowerPoint. We never used to get any of that stuff. I mean, when I was in university, very few professors even used PowerPoint. They used to use overheads. I mean, I'm kind of giving away my age here. But anyway, so the way that people learn things in study has changed over the years. And I think you're all part of a very large, uncontrolled experiment that is figuring out the best way to do these things. But at any rate, if you have the time, it certainly wouldn't hurt you to go back and listen to the lectures again, especially ones where you found them to be confusing, or maybe you didn't pay attention, or maybe you weren't able to give them your full attention the very first time around, and things along those lines. If you happen to have time for that, that would be good. Otherwise, I would really try to take the hints I've given you, take what you can find from reviewing the content, and try to just go through and invent pseudo questions of a sort that match my style. So think about questions that I could ask centered on these themes. Think about how you would answer them. It's also helpful to sort of index your notes so that you know what slides they came from and what topics are covered in each class. You have the slides available to you as you're writing, so the more indexed they are, the better you will be able to find the information that you need in a pinch. And yeah, so good luck with all of that. Of course, let me know if you have any questions about the material or anything along those lines. As I mentioned previously, we will be offering a chance to sort of improve your overall mark based on the performance of your final. So what I'm going to do is reallocate your marks as follows. If doing so would improve your final grade, I will reweight the marks such that the value of your lowest midterm is transferred into your final exam. So normally your final exam is worth 30 and each of your 2 midterms is worth 20. Suppose you did rather poorly on the first midterm, which many people weren't super happy with their first midterm mark. That's understandable. If your final mark happened to be better than your midterm, or one of your 2 midterms, what we'll do is scratch out the lower of the 2 midterms and then take the 20% that it's worth and add it into the final exam, thus sort of allowing you to spread out your better mark across the course more and recover your final grade a little bit if you happen to have done poorly in the first exam. Now, of course, if you do worse on your final exam than either of your 2 midterms, then we just keep the original grading scheme. But if it would improve, we'll do this reweighting. And this will all be done automatically behind the scenes. So what you see on Brightspace isn't going to reflect necessarily what you get on Carleton Central when your final grades go in. The Brightspace thing is really just to kind of track your progress as you go. It doesn't reflect your final grades. I'll put that all through I'll calculate that and upload that to Carleton Central separately. So I would say, like, this is a good incentive to try extra hard to study for this exam. I'm not making an easy exam. I don't believe that I ever have made an easy exam. So I'm not saying that you're going to get necessarily benefit from this arrangement. You'll have to put in the work and actually do a lot of studying and get ready for this exam and take it seriously in order to be assured that your mark is actually going to go up and this arrangement will benefit you. However, I've seen people's marks go up from the first midterm to the second midterm, and this wasn't because of some kind of like a curve that I applied to the grade. I do curve the grades with a sort of a pretty basic formula, but they're always just normalized to whoever got the highest mark in the class. So the fact that everyone's grade went up means that even before curving it, everyone's grade was actually still higher than what they got in the first midterm before curving that one. So you really are doing better in this course. It's not some kind of mathematical trick that I'm applying, although the mathematical tricks do help a little bit here and there, but that's just, marks are kind of fake anyway, so whatever. Okay, so let's talk about the meat of this lecture. We'll get through the content, and then I'll get out of your hair, and we can be on with our days. There's just a few social topics here that I want to discuss, because genomics research is, as I've said, so socially relevant, because we all have DNA, and like it or not, the impact of this research is going to be felt in our day-to-day lives as we go forward. And anytime you have a situation like that, you always have to ask whether the research, which affects all of us, whether we like it or not, is going to help society by making it a more fair and equal place, making it a more just place, or is it going to hurt society by reinforcing existing hierarchies and injustices? And I guess the extent to which you would really endorse that question depends on how good you think society is at the moment. If you think society is hunky-dory right now, and genomics research just solidifies that, then that would probably be good news from your point of view. But if you think there are some problems with society, genomics research is not going to, and genomics research is promising to solidify these problems, that's not going to be a good thing. But that's not for me to say. That's your own sort of decision. I'm just going to tell you what I suspect genomics research is going to do. There are many ethical and policy issues. So, for example, direct-to-consumer genetic testing like 23andMe and Ancestry.ca. We've used data from 23andMe a fair bit in this course because they are like a gigantic resource for SNP genotyping. These companies maintain ownership of your data. So when you submit your DNA to 23andMe, the genotyping data that they get belongs to them. It doesn't belong to the public. It doesn't belong to you. It doesn't belong to the government or to anybody else other than this company. And the company is run as a private concern that doesn't really have, it operates outside the realm of democracy, let's say. You can complain a good deal about the government, but the only, one thing you can say about it, at least, is democratically decided. So if there's a real problem, you could, in principle, vote somebody else in and have some sort of say over it. But if you have a problem with the CEO of 23andMe or Tesla, sorry, but you can't vote Elon Musk out of his own company. You can hassle him on Twitter, but you can't actually do anything to hurt him. So there is a bit of a big difference, or there is a difference there between private and public concerns. But anyway, these companies maintain ownership, and they don't really need to ask you every time they share it. They'll ask you sort of broadly, are you okay with sharing it? And if you say no, then in principle, they wouldn't necessarily have to share it. But when it comes to the individual uses of your data, you don't have any say about that. They can use your data for pretty much whatever they want without your knowledge. So as an example, or I guess rather an interesting case study in this, is the story of this fella called the Golden State Killer, who was active in California from 1976 to 1986. So a 10-year serial killer. It seems like the 70s and 80s were really the heyday of serial killers. You never hear about serial killers anymore, but back in those days there were a lot of these guys, and so he was at large for 10 years and killed many, many people, but he was not caught until 2018, after police took DNA that they knew belonged to him. They had DNA from the crime scene, I guess, and they submitted it to GEDmatch, which is a genetic genealogy service. So this kind of puts you in the position of the police here. You have DNA from somebody you think is the killer, but if you can't match that person's DNA to somebody in your database, how on earth are you going to use that to track them down? If the Golden State Killer is some kind of genius serial killer, he can cover his tracks very well and maybe never be caught. There's no public database that apart from databases of known criminals or known convicts or anybody else the police happen to pick up and hassle. Outside of those people, if you've had no contact with law enforcement, your DNA is not really going to be on file. So how do you find somebody? Well, the Golden State Killer himself was probably smart enough to not submit his DNA to genealogy.com or to ancestry.ca or 23andMe. I mean, if you have the DNA of a murderer, you're probably going to want to make sure nobody else can see your DNA. However, the beauty of this is that they don't need his DNA. They just need people in his family, because the genotyping services give you enough, or there's enough information in your DNA, and there's enough data on those genotyping services to be able to figure out if this guy has any brothers, sisters, cousins, grandparents, third cousins, anybody else in his family that is even remotely linked to him, will come up as a match in this service because of just the inheritance structure of these variants. So he doesn't need to submit his own data. It works even if your third cousins have submitted the data. So the police were able to find this guy because they took his data and found his, I guess, cousin or something. And then by looking, now you know what family the killer's in, you can study the family tree and say, okay, hey, do you know anyone in your family who might be a murderer? And then the family members would say, oh yeah, Joseph D'Angelo, he probably is a murderer or whatever. Like, once you have the family narrowed down, you can do a lot of regular just detective work and figure out who might be responsible. So what we're saying here is that, in a sense, you personally don't even really have the ability to consent to these services because you're involved in them, even if you personally keep your DNA completely private, because people in your family who you can't control, the act of them submitting their data puts you into the system implicitly just because of the inheritance structure of DNA. If anybody in your family is in the system, or if multiple people are in the system, your DNA can be linked to them very easily through these databases. So there's a big risk here. Now, I don't think anyone's going to complain about catching serial killers. I think that's probably a good thing. But there's lots of other ways this could be used maliciously. Think about the potential risk of having this data available to insurance companies. Think about the potential risk of having this data available to employees, or to the government, or to the police if you're not a criminal, or if you're whatever. I mean, like there's all kinds of people who you don't want to have this data, but you don't really get a say in it because the data is owned by private companies, and because people who aren't you can submit data that tells a lot about you, whether you like it or not. So there's really kind of uncertainty about all of this. There's also uncertainty what happens to the data if the company that owns it goes bankrupt and decides to sell off their assets. The DNA data is an asset, and what happens to it after the company dissolves is kind of an open question. So these are things to consider. I don't know what to, like I say, these I'm not, these are the issues you can think about it what you like. So I'll just give a few statements from an article that was published recently asking leading geneticists and genomicists about their opinions for what the future of genomics has to be. So Amy McGuire, who is a professor in genomics, writes, "The genomics of the future must be genomics for all, regardless of ethnicity, geography, or ability to pay." So this kind of gets back at a theme that I've given you a few times through the course, which is the overwhelming predominance of people of white European descent in the genomics literature. Most of the data that I've shown you, asking about the genetics of a given mental illness, for example, is coming from GWAS studies, genome-wide association studies. And of the 2,500 or so studies in the NHGRI GWAS catalog, the National Institutes of Health Genomic Record Initiative, their catalog, 81% of these studies focus on individuals of European descent. And of the remaining minorities, only 5% come from non-Asian minority populations, meaning that a vanishingly, most of the world's people, let's just say, are not represented by these studies that claim to be able to elucidate the genetics of diseases that affect everybody. So these disparities directly affect clinical practice because, as I've shown here and there, genotype can be used to predict drug effects. Genotype can be used to understand biological mechanisms, and this leads to changes how conditions are diagnosed and treated. And of course, if you're using information that's specific to one population group, you're leaving a whole bunch of people behind. So there is some progress on this. Funding agencies are increasingly starting to invest efforts to increase diversity in genomics research, but it's a long process. And of course, with money, there's always the question of where it's going to be coming from. One example by the way, of the way that this kind of plays out in clinical practice, Armstrong et al. In 2005 showed that African American women are less likely than white women to obtain BRCA1/2 testing, even if they had a family history of breast cancer. The BRCA gene, if you recall, was discovered by linkage studies of inherited or familial breast cancer. And it's a very easy test to have. They just look at your DNA for this gene, and if you have the bad version of it, your risk of breast cancer is quite high, and you might take precautions against it, whether it's a sort of prophylactic mastectomy or just keeping a closer eye on things or whatever it is you decide to do. So it's a pretty harmless test to take. And really, I mean, if you have any thought of risk of breast cancer, you probably should have it at some point. However, it just happens to be that this is not applied evenly through all the populations. People of African descent, for example, for whatever reason, are either offered the test less or choose to take it less, or if it's the case of a place with like in the US, they may just not be able to afford it or not want to pay the money for it, or whatever the case may be. This data, which should benefit everybody, is not being applied equally. And I think this is a problem, socially speaking. We would like to see the work of scientists applied fairly to everybody on Earth, but it's also a problem strictly from a scientific perspective. So even if you don't find the social issues terribly compelling, or if you think that they might be overblown, or whatever your thoughts on that would be, there's still benefit to pursuing this because the very nature of our genomes does differ depending on the population groups you look at. So if you recall, linkage disequilibrium is a construct that describes the extent to which SNPs, or different alleles, I should say, of SNPs are correlated with each other with regards to space, like spatial location on the genome. So SNPs that are in linkage disequilibrium with each other tend to be inherited together on the same haplotype block. During recombination in meiosis, these regions travel together on the recombinant fragment and thus are inherited together. And these fragments aren't random. These haplotype blocks are divided by specific boundaries called breakpoints that are kind of repeated or are kind of universal between people. So these breakpoints exist, they're in the same place for pretty much everybody. But they're in the same place for pretty much everybody of the same ethnic background. So, for example, in European people, the breakpoints are in one place, and so the linkage disequilibrium structure is one way. But in people, say, of African descent or East Asian descent, the breakpoints can be in different places, and thus the overall linkage disequilibrium structure can vary. The overall level of genetic diversity, for example, is higher among African populations, and as a consequence, the overall linkage disequilibrium levels are lower. This means that there's less, like, if you think about the ability to impute genetic variants from index SNPs that we talked about in the Alzheimer's lecture, the neurodegenerative disease lecture, that ability is lowered in highly diverse populations with low levels of linkage disequilibrium, and that would be like African populations. So this linkage structure really affects the ability of us, our ability to do genome-wide research in an objective fashion. Yet the groups of people are oftentimes treated as though they're all fairly similar. What's more, as I've said, there is a fairly sizable overrepresentation of people of European ancestry in polygenic risk studies. So here's a plot that I showed earlier in the year. The x-axis is years when the studies were published. This goes up to 2017. The y-axis is the cumulative number of studies. And then these are genome-wide association studies. And the color represents the population group that was studied in those studies. So you can see that the blue bar, the people of European ancestry, make up the majority of those subjects studied. The next after that would be Asian, and in particular East Asian people, Japan, China, and Korea, these kinds of places. Mixtures of groups are represented by gray, and then this microscopic little area here would be everyone of African, Middle Eastern, or Latino/Native American origin. So these people represented by this smaller block make up a very large percentage of the world's genetic diversity and a very large percentage of the world's overall population, yet are dramatically underrepresented by the genome-wide studies that purport to be able to discover the origin of diseases. And as a consequence, polygenic risk scores, which are able to use your genetic data to predict your risk of a given condition, whether it's breast cancer or schizophrenia or any other kind of cancer or whatever else, even phenotypic traits like say stature or body mass index, or whether or not you can I don't know how good you are at singing, whatever kind of things that they look at. Those tests all perform worse when they're done outside of the reference population. So the tests work well in people of European descent. One means that's like the baseline. They work fine. And then people of like American, and here they're talking about sort of people from the American continent, Latino, Hispanic, and American Indian type people, the tests perform worse. They perform, about 60% as well. And all the way down here to people of African descent from the continent perform less than 25% as well as the same tests do in European populations. And again, we're talking about a sizable portion of the Earth who are underserved by this work. So I think this is interesting both from a social and a scientific perspective, because we continually find ourselves at a loss to explain the heritability, the apparent heritability, I should say, of a variety of conditions. We do these genome-wide studies and find that we can explain, maybe 7 or 8% of the variance in a given, in risk for something like some kind of mental illness, and yet much of the world's genetic diversity remains unexplored, or at least underexplored. And this diversity actually might be part of the secret to finding more of what's going on. Due to various sort of higher level topics like the lower levels of LD among African populations, for example, it makes it easier to find map traits, meaning that it'll be there's more chance of finding the variants of interest in those groups potentially. So I don't know. I think this is just a kind of an interesting sort of a point. It's typical, I suppose. Well, I don't know. I think it's funny that universities and funding agencies both really want to give the appearance that they're willing to combat inequality, I guess, whether it's material inequality or social inequality. And if you apply for jobs at these places, they'll oftentimes make you or ask you to write a statement to that effect. But it seems as though if they really want to make a material difference here, what's actually needed are resources. You have to actually be willing to spend the money to get these data that we clearly need. And I hope I've made the case that we would be benefiting if we had that data. When it comes to actually spending the money, these places often fall short in doing it. They always sort of tend to go for the easier and the more tractable approach. Obviously, if you live in Northern Europe or in America, where most of the money is, it is easier to reach out to people of European descent, not only because their genetics are better understood and thus it's easier to integrate your data into the broader data set, but also just because that's where the people are and that's where you are. So why not? I think, ideally what we'd like to see is genetics research led by PIs outside of the existing infrastructure, people working in their local communities, places like sub-Saharan Africa and Southeast Asia and other kinds of places that are relatively underserved by this type of research at the moment. Science, is sort of the birthright of all humanity, and so it doesn't really seem right to have it so concentrated in one part of the world, if possible. And when it comes to solving these problems and getting genetic data on more diverse populations, it also doesn't really seem appropriate to have a bunch of American or Canadian or British PIs parachute into a place, grab all the valuable data, and then teleport, publish it in Nature or Science or some kind of very fancy journal, and then teleport to their comfortable positions at, their tenured positions at Columbia or McGill or something. That's kind of colonial almost. Rather, we'd like to see local expertise fostered so that developing countries can build their own R&D infrastructure instead of being used as a resource by the rest of the world, which is, of course, something that they're already used to experiencing. So I think this is kind of, it's just funny, I guess, to think about this dichotomy between the stated values of diversity and equality that most universities would claim, up to the point of being considered like havens of like far-left communists or whatever. Really, universities are rather reactionary places that tend to be quite conservative about their budgets and how they spend money. It's much more important to look good and say the right things than it is to actually use up your resources trying to solve the problems that exist in the world. So people who get angry at it, it's a bit like if you watch like a wrestling, wrestling, and you yell at the characters on the screen as if it's not all kayfabe. But anyway, that's my rant of the day. I can only say that because we're in the last class, but I think I hopefully I've made my point. And this is the class about social issues, so it's something to consider. And really, it's not only about our sense of justice, because of course justice is not a topic that is handed down for us to look at universally. It's a matter of resource allocation, but there are benefits to it aside from just that side of things. Looking at diverse populations can allow for new discoveries. You can find things that you wouldn't have been able to find if you'd only focused on one subset of the world's population. So, for example, it was noticed back in 2005 that African Americans with a nonsense mutation in the gene PCSK9 have lower levels of LDL cholesterol. So that's what this is showing. LDL cholesterol is sort of the bad cholesterol. Low-density lipoproteins are these sort of like cholesterol-carrying entities that are somehow bad for you in some kind of way. And so when you talk about like cholesterol levels, these are the ones you don't want to have high levels of. So drugs like Lipitor and things like that are aiming to kind of reduce your levels of LDL or improve the ratio of HDL to LDL. But anyway, here's a histogram showing the distribution of LDL cholesterol levels in people with a normal copy of PCSK9. And you can see the mean is around 125. And then in people who happen to have this mutant version of PCSK9, their average was below the 50th percentile, or below the median of people with the normal copy. Now, this is a mutation or a variant that was unique to the African American population that was studied in this example, but it led to a rather important set of clinical discoveries, which is the development of anti-PCSK9 drugs as treatments for cholesterol, because the mutant version of this gene doesn't work in these people, and as a consequence, they have lower cholesterol and they're otherwise fine. So the thinking would be, if you can develop a drug that targets this enzyme, then you would be able to recreate that cholesterol lowering effect even in people who have a genetically normal copy of this. So this is just one kind of example why increasing the range of population studied benefits all of us, because you can discover new biological mechanisms that hadn't been conceived of before. So, if you see a real life example of somebody with a messed up version of PCSK9 and see that it lowers cholesterol, that gives you a very valuable direction to go for your drug discovery. Now, ironically, these drugs that work on this principle remain expensive and poorly covered by insurance companies relative to statins. So, Lipitor would be a statin, for example, but that's a separate kind of question, and I think we could all have one or two complaints to be making about insurance companies. But be that as it may, the benefits I think are still there. Now let's move on to a different part of this topic. This is more getting into the future directions for GWAS research. So here we're looking at a plot of time. The first GWAS studies started to come out in late 2007, early 2008, and they've been continuing to this day. Although this paper cuts off at 2017, the trend continues. So that's the time on the x-axis. And then over here on the y-axis, we have the cumulative number of SNPs that have been linked to various traits and conditions. And you can see that as time goes on, the studies get bigger and bigger and bigger, and more SNPs become discovered. So you have years where certain highlights are made. For example, Alzheimer's disease and age of onset, glomerular filtration rate, educational attainment, and all kinds of other things. GWAS studies, by the way, are used for all different types of topics, not just mental illnesses and whatnot. Everything you could dream of has been done in the format of a GWAS study. And as time goes on, more and more trait-associated SNPs become associated. And what this seems to suggest is that if we keep getting bigger and bigger studies and keep doing more and more of them, we'll find more and more trait-associated SNPs. And you never know which SNP might happen to be the one that really gives you a deeper insight. Sometimes a SNP, as I think I've implied, might on its own be rather trivial, but if it's located in a gene that's very informative about the underlying molecular biology of the condition itself, then the GWAS study can lead you to some rather interesting biological insights. So this is also something that we can look forward to in the future, although for many conditions, we're getting to a point where we can't make the studies any bigger because there just aren't enough people with that condition on Earth, at least within the current population sets. As I've said, it would be very beneficial to explore the rest of the world outside of just the US and Northern Europe. I guess I should say genetic correlation does help with this as well. As more and more of these traits become discovered, it's possible to make more and more correlations between more and more genetic correlations. And that has the ability to take a weak study that has a small sample size and kind of chain it onto a bigger study with a bigger sample size and use those correlated SNPs to kind of bootstrap or to improve the power of the weaker study. So, for example, a famous or one that comes to mind is the fact that the genetics of anorexia nervosa and BMI are correlated. And BMI can be studied in hundreds of thousands of people. Anorexia nervosa is much harder to study because it's not as common. So you can get maybe hundreds or tens of thousands or thousands of people in that kind of study. But by showing that there's a correlation between the variants, you do now have an insight that the genes that are related to anorexia nervosa probably are similar, maybe the same sort of biological pathways as those that control body weight in general, which is a rather interesting finding if you ask me. That, maybe it seems obvious, but you don't know until you do the research. Now, we can also look at how GWAS hits can be linked to drug development. That certainly does happen. So, for example, this figure here shows how GWAS hits, genes located with or genes with SNPs that linked to diseases, lead you to genes, and then genes, because they're part of known biochemical pathways, can lead you to drugs. So there's a few examples given here. For example, in our world, schizophrenia is one of the genes that comes up in a GWAS is DRD2, that's a dopamine receptor. And antipsychotic drugs are typically blocking dopamine receptors. So there is this link between the genetics of a given disease, the genetics of a given disease and the drugs that we use to treat it. And the same could be said for like type 2 diabetes or rheumatoid arthritis or various other conditions. But more often than not, the genes you find in GWAS studies don't have an obvious link to the biology of the trait that you can drug. They might make sense in some sort of abstract way. Many of these conditions, for example, involve genes linked to neurodevelopment, but they don't necessarily have a druggable target. If you have a gene, for example, that's involved in like axon pathfinding, which many of these genes are, you know that it probably has something to do with how the brain wires itself up and how the axons extend and find their targets during brain development. But that's not something you can exactly drug. For one thing, if it concerns a condition that's diagnosed in adulthood, the damage is already done at that point. It's already manifested as a structural difference in the brain. You can't use a drug to fix your axons and where the axons are located. So it puts you in a rather difficult position in that regard. But it does seem like this kind of thing could lead in a good direction if we use GWAS studies to estimate drug effects, so that perhaps someday you might be able to be given say an antidepressant on the basis of your genetics, and maybe make it less of a trial and error process getting people hooked up with the right meds for their condition. That's what we would like to see anyway. Who knows if it'll happen. So as I've hinted, most of the complex multifactorial traits we see arise from common variants with relatively small allelic differences at thousands of sites across the genome. What this means is that wherever possible, GWAS studies have to continue to get bigger and more diverse to identify all of them. But even now, for the big studies, effect sizes can be extremely small and still significant. So we see studies, for example, where the odds ratio of a given allele or a given variant is like as low as 1.01. That means that having the risk allele, having the bad version of that SNP, increases your risk of a given condition by about 1%. Now that's a ludicrously small difference. Would be completely uninterpretable on its own. But when combined with your genotype at many other variants, you can develop polygenic risk scores that put all that together and give you a sense of your overall risk. It's a bit like, there's a lot of things in your life that sort of somewhat contribute to your likelihood of a certain outcome. For example, if you're thinking about what your risk is of passing an exam in university, you can take a lot of factors into play, like how much did you study? How well did you sleep the night before? How well did you understand the material? The day of the exam, did you have an easy ride to school or was it a tough bus ride? Did you get enough coffee in the morning? Did you have too much? There's all kinds of factors that contribute a tiny little bit. Did you get out on the wrong side of the bed? Did your cat wake you up by puking on your shoes? These kinds of things all factor into it. And on their own, they would each make a fairly small difference in the outcome. But when you stack them all up, you can end up with either, they can end up swinging things in rather large ways. This is the essence of what a polygenic risk score is. So they're really just trying to do that type of analysis across the genome for your with your risk of a given condition in consideration. And these variants that we see with GWAS studies tend to occur in regulatory regions. Think back to our functional genomics lecture about enhancers and promoters and these kinds of things and transcription factor binding sites. These things are not genes themselves precisely. They're not coding for proteins, but they are changing the way that genes are expressed and in what tissues and at what time points and things along those lines. So these regulatory networks end up being rather important for understanding the results of GWAS studies. And they'll be needed to help us understand complex traits. We also have more work to do when it comes to unraveling the layers of the epigenome. The idea that DNA methylation can regulate gene expression dates back to 1975, so it's not exactly a new idea. It's older than I am, believe it or not. But the concept of histone modifications as regulators only dates back to the year 2000. So that's a comparatively new idea. Although I suppose it might still be older than many of you. But it's not, to somebody like me, it's not that new of an idea. But these are fairly, compared to what we know about DNA as a mediator of heritability, these are fairly new. But we've made enormous progress in gathering epigenomic data. But we're still missing a lot of key pieces of information. For example, how is DNA methylation directed to specific sites in the genome? This is quite important. I think I've given you the impression, perhaps, that genes just are or aren't methylated, and the way that they acquire their methylation is left unsaid. That's unfortunately, that sort of matches our state of knowledge. We know that DNA methylation is associated with gene expression, such that genes with a high level of methylation tend to be lower expressed, and genes with low levels of methylation tend to be expressed at higher rates, and this varies between cell types and tissues. But where does the methylation come from in the beginning? How does a cell know that a given gene is going to be expressed at a high or low level? How does a cell know that it wants to shut off a gene using DNA methylation? Where does that information come from? It's a bit of a chicken and egg thing. What comes first, the methylation or the expression? Is it the case that genes with high levels of expression somehow resist DNA methylation being added? Or is it the case that, and it's the case, for example, that silent genes that aren't being expressed somehow attract DNA methylation? Or is it the case that the methylation is established first, and then on the basis of methylation, the gene either is or isn't expressed, right? This is kind of the question we have to ask, and it's a little bit tough to get at this. And then within that realm, how is the DNA methylation directed to specific sites? How does the methyltransferase enzyme know to find a given gene and start at the transcription start site and silence it there and everywhere else throughout the gene? How does it know? These are complicated questions that we're still looking for answers for. Right now, the thinking is that the expression comes first and the methylation comes after. But that is also variable because there seems to be kind of evidence for sort of both interpretations depending on exactly how you look. So we'll have to wait and see on that one. And then also, how did DNA methylation evolve? Vertebrates use DNA methylation quite extensively, but invertebrates, which make up most of the world, and don't even get me started on plants and fungi and all the other kinds of living things out there. Why is it that vertebrates seem to use DNA methylation so much? It's like, if DNA methylation is so important, why is it only present in vertebrates? DNA is present in every living thing. RNA is present in every living thing. Mitochondria are present in every living thing. Why is it that DNA methylation, which apparently is so important that we have to have it, if something messes it up like Rett syndrome, then you're rather severely impaired? How come that seems to be the case for vertebrates, but other animals or other invertebrates and other life forms don't necessarily need it to the same degree? Other life forms also use it differently. I mean, vertebrates do as well, but some insects use DNA methylation, but in different contexts. Some plants will use DNA methylation, but in different contexts. So it's a thing that exists and may have evolved a number of times. It's even present in some archaea and bacteria. So it's been evolved a number of times. It exists throughout the tree of life, but creatures use it in different ways, and it certainly doesn't seem essential for life in general the same way that mitochondria and DNA happen to be. So, that's a pretty big question as well. And then another limitation is that most epigenetic studies are descriptive in nature. We are able to, with increasing levels of precision, describe where epigenetic marks happen, what tissue, what time point, etc. But to go in and change those marks in that specific tissue at that specific time is much, much more challenging. So we have a lot of data that's that gives us correlations, but not as much as you'd think about causation. It's very tough to go in there and edit the epigenome. It's easier to edit the DNA, by the way. I mean, the DNA can be edited reasonably easily these days using CRISPR and whatnot, but editing the epigenome is still quite a challenge. Let's get into another topic. We've talked about how the world of genomics research has gone through a number of phases. One of the phases that we saw prior to the genomics era would be linkage studies, where you're looking at pedigrees and family trees and people with a known level of genetic connection, and you're following given traits or phenotypes through the family tree and trying to find markers in the genome that follow or that appear to be linked to that condition. And then you can use the location of those markers to guess where the genes might be located. Now, we've really liked GWAS studies because they take away this requirement to have a well-phenotyped family tree and now let you use giant numbers of unrelated people. This work has given us a decent sense about how common-ish variants contribute to your phenotype and to your disease risk. So GWAS studies, of course, we know are quite useful and quite valuable. But we are now beginning to sort of hit the limit for where they can be. As I've stressed, we do need larger sample sizes, but sometimes you just can't get any bigger than what you already have. The sample sizes are as big as they're probably ever going to realistically get unless you start moving into different population groups that are outside of the normal sort of white European groups that we've seen previously, and I've made the case for that. But we also have to look in other places for our causality, and we're seeing now begin to sort of a return to studies of families, where we are bringing back pedigrees, folks. So a very recent study comes from Wilford et al., published just this year, in fact just a month or two ago, who found that ASD, autism spectrum disorders, are associated with what they call private variants. These are variants that are recently evolved and unique to families. They are variants so if you have ASD and it runs in your family, and you could look at a pedigree and see that certain people of your parents and grandparents' generations have also developed ASD, you have a situation where it's inherited. Now, when I was talking about ASD, I kind of gave you the sense that there are two big bins that you can put the genetics of ASD. One bin would be the heritable form that was that you can find using GWAS studies. So these are relatively common variants that have a effect in the general population. So you've got the things you can find with GWAS studies that are heritable because they're common enough that you can genotype them and they are widely spread in the population and correlate with the disease. So you've got the GWAS variants. Then you've also got the de novo variants that, as I said, are brand new. They don't exist in the general population, so normal genotyping studies can't find them, but they also play a role in ASD because you see an enrichment for de novo variants in people with ASD. These would be tandem repeat expansions as well as just straight up de novo mutations. So you've got those two things. But there's a third kind of thing that only now we're beginning to be able to find, which are what they call private variants. These are differences that exist within a family that you don't really see in the general population. So they're individual mutations that wouldn't be picked up using normal GWAS methods, but you can find, if you sequence the DNA from family trees, you can find these variants that travel with ASD and link the variants to that. So using methods that are rather similar to these old linkage studies or these old pedigree-based studies and whole genome sequencing, not SNP arrays, but whole genome sequencing, you're able to find this third category of things that are extremely rare but heritable and not de novo mutations. They're not de novo because you inherit them from your parents. So this is a private variant, and this is what Wilford et al. Describes in the case of ASD. This is just a picture from the study. You don't need to worry about too much what it says. It's just an illustration of the findings. But what's cool about these is that they are recently evolved. Because you have a family tree, you can go back through the whole family and say, okay, well, this is a variant that is linked to ASD. Where did it come from? When did it appear? And on the basis of a number of sort of complicated computational methods, they're able to calculate that these variants are only about 2.5 generations old. So they're very, very recent in the evolutionary history of human beings. And likely, if you can infer from that they're under some kind of purifying selection. So they don't last very long. They'll eventually be filtered out of the gene pool as anything would that doesn't help your survival. So I'm not trying to, I don't want you to, worry too much about explaining to me exactly what a private variant is. I'm just throwing this in at the very end of class to tell you that in addition to all the things I've given you in this class, there's a whole bunch of other stuff that's out there coming soon that I haven't had time for, including this sort of return to pedigree studies. And this is a great thing because pedigrees, while they're more complicated to use, you have to go and find families and characterize them, and it's a lot more work. They do give you some advantages. For example, you have a known baseline allele frequency because you're working within a related family. You'll know the frequency of all the alleles in that family tree. They also account for the unique effects of rare variants that you wouldn't find in normal genotyping panels. So they kind of combine the advantages of GWAS studies and linkage studies. They're also better for rare conditions where you don't have hundreds of thousands of people you can look for. So something to look forward to there in the future. We're also seeing the way that genomics data is being integrated into medicine. As I've said, you can use GWAS data to make guesses about what drugs to develop or what drugs to use, but we have kind of a more systematized version of this, which they call the axes of health data. Many people in the healthcare space are very concerned with collecting more and more data. There's a thought now that what's really missing from our understanding of disease and health is data. This is why all of the big tech companies have gotten into collecting data with, smartwatches, and your phones will track how much you walk and all kinds of other vital statistics now. Health is a big part of what Apple's invested in, and Google, and all these other tech companies are big into health because they realize that, well, for one, health is a big moneymaker. Just look at the profits of insurance and drug companies. And number two, this is something that everyone cares about, and we're clearly lacking for data. So this sort of inclusion of genomics into medicine is the big goal here. And even though we have vast quantities of genomic data for many, many different conditions, the knowledge is really only applied in the case of certain cancers and some monogenic conditions. So the ratio of knowledge that we have to the application that we see in the clinic is pretty disappointing. We have a huge amount of knowledge, polygenic risk scores, and all these other kinds of pathway analyses and gene ontologies and whatnot, but very little of it has really had a strong effect on clinical practice, which is a rather disappointing thing. So we need to kind of work on that a little bit. We also need to ask questions that are a little bit more creative. For example, we can keep making our studies bigger, making them more refined, look at single cells, more time points, etc. But we also have bigger questions like, what are the roots of variation itself? Why is it that people are different from each other? That's a fairly big question. The genetics maybe don't answer everything, because identical twins can be different from each other. People can be different from each other. What explains most of the inter-individual variation? Is it genetic, or is it epigenetic, or what is it? And then how can we use genetics to generate actionable and personalized insights for health? This still remains to be done. Many countries are building their own biobanks. A biobank is little more than a giant database of genetic data and extensive phenotyping. So it's sort of valuable in a sense that, like, if you want to do a GWAS study of a given trait, whether it's height or eye color or a hair color or body mass index or singing ability or hearing ability or whatever, I'm just thinking of random things, but you can do a lot of stuff. You could get the money to do that, develop, assemble a team of geneticists and clinicians and whatnot, go find the people, do all the DNA analysis, do all the phenotyping, put all of this together, and it would cost you millions and millions of dollars, and it would be very tough for individual researchers to finance all of that. What's also possible is for nations to sponsor their own biobanks, where they will, where the government essentially will go through the work of finding the participants, paying for all the extensive phenotyping, getting their genetic data, getting all of their genotyping done, and putting it all on a database that's available publicly for researchers to use. This is a better approach in some sense because it takes the onus off of individual PIs and puts it onto larger and more powerful entities like states and health agencies. So for example, the biggest one, or the most famous one, is the UK Biobank. They've got data on 500,000 people. They have been extensively phenotyped and studied every which way, and we have their genetic data. So if you want to ask a question about the genetics of a given condition, the data from the UK Biobank could probably answer that, even if the authors or the people who worked on that biobank originally never planned on you asking that question. You can look at the data yourself, ask your own questions, and use it however you like. So that massively reduces your burden as an investigator for getting the money and putting together the study. The government was able to do a better job of that than you were. And there's similar efforts in Korea and in Japan and Taiwan and China and Canada. Of course, God love us, we also have a little biobank, 43,000 people that they're working on. So it's, every country is trying to join the fun. The U.S. of course has one that is, they have over 1 million people in their big biobanks. So, you're not going to beat them anytime soon. But everyone's doing their best because this is something that is seen to have a lot of potential. And by the way, the more of this kind of stuff that goes on, basically down in like this half of the world where you only really see white, uncolored countries that don't have biobanks, mostly in the global south. except for Australia. I don't know what's going on with those guys. But the rest of the world down here is severely lacking in this data. As I've said, this is something that does cost a lot of money and requires a lot of expertise. But this is just something that has to be worked on, I suppose. What else do we have? So some other thoughts about social responsibility and genomics. So there's a lot of conditions you can study that are fairly neutral, that describe basic aspects of human variation that aren't terribly value-laden. I mean, I guess everything is value-laden to a certain extent. I was going to say height isn't value-laden, but obviously people do ascribe a lot of importance to how tall you are. BMI, of course, has a lot of import in that same way. But there are other ones that have even darker history. For example, things like intelligence or criminal behavior. Educational attainment is sort of taken to be a measure of those things. Income, sexuality, these types of things. These traits are very likely to be influenced by non-genetic social factors, and they're also very likely to cause, I guess you might call them social problems, on the basis of the results. Because the world that we live in is unequal. There are inequalities in society, of course, and they break up along class lines and along racial lines and along many other lines that you can visibly observe. Now, if you take if you take those categories along which society is unequal, class or race or whatever the case may be, and look at the genetics of those things, you might find yourself in a position where your interpretation of the genetic data legitimizes those inequalities. If, for example, you take the bottom 50% of earners and the top 50% of earners and you look for the genetics of intelligence among them, you might discover, perhaps, that intelligence, whatever that means, is higher in the top 50% compared to the bottom 50%. I'm just throwing an example out there. And that would say, actually, the society's fine. It's just that, the smart people rise to the top and the people who aren't smart fall to the bottom. And that's proper, isn't it? That's very sort of nice Calvinist moralizing here. So this is always a risk with this kind of research, where you want to study the variance itself without saying whether or not the variance should be there. Philosophers would call this the naturalistic fallacy. Just because things are a certain way doesn't mean that's the proper way for things to be. But anyway, geneticists themselves would urge people that these things are worth studying. I mean, if intelligence is a real thing, you should probably understand the genetics of it. Similarly, if criminality or social deviance is a real thing, it's better to know about it than not, etc. But you can't be fatalistic about the results, and you have to resist the temptation to essentialize people on the basis of their genetics. But this advice is rarely listened to. So people have brought this up for many, many years. Of course, there's a very dark history with it in the first half of the 20th century with the eugenics movement and fascism and things along those lines. But it never really goes away. Even in 1994, there was a book called The Bell Curve, published by Richard Herrnstein and Charles Murray, that basically just made the point that race and class disparities were coming from immutable differences in inherited intelligence, not any sort of social factors that might be under our control. And that's a very convenient belief. If you think that differences in outcome or inequality are just genetics, well, we can just throw up our hands and say it's all genetics, folks. We can't do anything about it. Society works exactly as it should. So this is important stuff, because it's, you can't, if you're doing the work, you can't say it's definitely not genetics. You also can't say it definitely is genetics. You have to think of a way to study it and it's not biased. And that type of study is very hard to do, potentially even impossible, because we can't separate our bias from ourselves, or, we can't separate our research completely from bias. So it puts us all in a very awkward position of trying to find the truth, but being aware that not only are we tremendously biased by the way our society is set up, but also the results we find might actually make the problem worse, depending on what it is we find and how we interpret the data. So there's a lot of pressure on researchers in many areas here that I can't really speak to, but I guess you should just be aware of it. So as an example, something that may surprise you is that there is a fairly sizable genetic basis for educational attainment. Educational attainment is simply the number of years of schooling completed. Maybe you have 5 years of school you've done. Maybe you have 15 or 20 or God knows how many years some of us have been in school. But it's just a number that you can measure really easily, and then you can do a GWAS study with that number as your dependent variable. So it does appear to have a genetic basis, as surprising as that might be, when you think about all the social factors that could impinge upon it. But it's been subjected to a very large study of 293,000, and that's quite big. That's big enough where you could start to think that you would actually be able to control for a lot of the confounds. And here's the Manhattan plot from it. They found a number of SNPs that are linked to educational attainment. And further supporting the kind of biological reality of all of this is the fact that the genes they find tend to be in neurodevelopmental pathways. That's what this shows here. This is a pathway analysis. So you see terms like dendritic spine organization, cerebral cortex morphology, synapse organization, voltage-gated calcium channels, dendrites, and so on and so forth. So these are all things that have to do with brain development, and at some level, I guess it's not surprising if you have a study of educational attainment. Probably the brain is the most relevant organ for that, and the most relevant process for that would be the development of the brain. So you do have a story that's biologically plausible, and that does seem to add credibility to the fact that you haven't discovered a spurious signal. If the genetics of educational attainment were based purely on biases that exist in your data set, then I don't think you would necessarily expect to find so many brain-related genes, given the plausible connection between the brain and educational attainment. You might expect to find random genes having to do with kidneys or liver or muscles or something like that. But who knows? Not necessarily, I suppose. But anyway, this is all just to say that, like, having this data in your hand puts you in a very awkward position. Is your ability to perform in school determined by your genetics to any severe degree? Do your genetics affect brain development and thus your ability to achieve in life? It depends on the way your brain develops, and that depends in large part on your genetics. If that's true, that's a pretty big finding. So you have to be very certain it's true. And I'm not saying it is or it isn't. I'm just saying that, like, this is knowledge that exists in the world, and you have to really reflect on what it means and what you would do with it if you were in the position of either discovering it or commenting on it as a member of the public. So big topics, really, if you think about it. But we must urge you not to be fatalistic about it. No matter what your genes say, they only describe your approximate probability of something. Remember, even the best GWAS studies only explain a very tiny fraction of the variation. And what's more, the genetics play out differently in different societies. The nature of the society influences how differences in genetics manifest. So, for example, Rimfield et al. Back in 2018 showed, on the basis of twin studies, that the genetic differences, or the genetic contribution to educational attainment explains twice as much of the variation in post-Soviet Estonia than Soviet Estonia. So the y-axis of this figure represents the proportion of variance explained by genetics. This is on the basis of heritability, like classical twin studies. So there's a genetic component and two different kinds of environmental component, and the genetic component is what's being measured here. And so the higher the bar is, the more your genetics explains your difference in outcome. And if you look at people who were born during the Soviet era, and Estonia, by the way, exists in an interesting place in the world. So early in the 20th century, it was under Russia's influence in various ways. But after the October Revolution and after the Bolsheviks took over, it became part of sort of Soviet influence as part of the secret terms of the Molotov-Ribbentrop Pact, which is rather interesting. See, like, these countries on the border between big powers always end up being kind of passed back and forth and going through these conflicts. So it was used as sort of a secret part of the Molotov-Ribbentrop Pact. And as a consequence, Soviet troops were gradually moved in and began to occupy the country. It was annexed effectively in 1940. And it was briefly taken over by Germans in World War II, of course, and at the time, people basically had a choice between fascism and communism, which is not an unusual dichotomy. Usually when one is on offer, the other is also, at least historically speaking, the two things tend to arise kind of in reaction to each other. But anyways, eventually after the war, the Soviets reoccupied the country, and this lasted up until Perestroika, when the Soviet Union began to liberalize, and it gradually sort of fell apart. So after the fall of the USSR, Estonia was integrated into the neoliberal world order and subjected to the usual type of neoliberal shock therapy. You can read Naomi Klein's book about that if you like. But anyway, suffice it to say, it has an excellent education system. And what I'm getting at basically is that after this Soviet era ended, people's genetic endowments had a stronger effect on their educational outcomes, which means that, I guess it's implied that the sort of forced equality of the Soviet era meant that it was holding people down, because even if you had genes to be a genius, you weren't allowed to go any further in school than the central planners would let you go. Now, it's funny, I think, in reading this paper, the researchers compare their results to the UK, which they describe as a true meritocracy, which I think is a very funny thing to say about a country with a literal royal family. But anyway, this is a message, really, I think, about how differences in your genetics and your ability only matter in the context of a society that enables those differences to rise to the top. That means we can theoretically shape a society that's less sensitive to your endowment, but still fair to people who are able to excel, at least in principle. Another kind of interesting thing is that you can link the polygenic score for a child's educational ability, I guess, and show that it affects parenting strategy, but only in parents that don't have a college degree. So this is educational attainment genetics, and we're looking at the effect that educational attainment polygenic score has on parents' help with reading and so forth. So I guess I don't really have a lot of time to get through this. But anyway, the point is that the genetics of the child affect how the child is parented by their own parents. Children with a genetic propensity toward higher educational attainment actually get more attention from their parents, but this effect is especially true in parents that don't have a university degree. Parents that do have a university degree invest huge amounts of effort in their children either way. So this is a rather interesting thing. I think it has to do with the psychology of the middle class and how they're especially concerned with this sort of precarity they find themselves in. People with university educations are very aware or very concerned with the possibility, or I guess with the role of effort in their level of achievement. So they're especially fixated on making sure their child is getting everything they possibly can have. Who knows, really, I guess, but it's just another example of how genetics and nurture interact. The nature and the nurture interact with each other, and it's very difficult to say, if you look at this from the big picture, what is really playing a role, because the child's genetics obviously is taken to represent something, but then the parents look at this, or the parents look at the child's own performance, and that stimulates their own investment in the child. So it's kind of a complicated back and forth. So we'll just close off with some thoughts from researchers working in this space. I won't read through all of these, but you can go and look at this slide. Marchenko in 2021 published just a series of interviews with people working in sociogenomics studying intelligence and educational attainment. Researchers are quite careful to point out their moral, their kind of responsibilities here. So one attitude, for example, from Carter says, because we're not controlling for genetic differences, it's going to be more difficult to find the true effects of those kinds of things. She's talking about the difference, like different styles of schooling, different educational interventions. You have to know, or it's helpful, I guess, to know about the baseline differences in genotype that affect educational attainment, so you can have something to use as a covariate in your analysis. Let's see. So there are no genes for anything. These are the researchers talking about their concerns with how the research is reflected in the literature. I feel very strongly that the onus is on us to educate people and to explain very carefully what we mean when we talk about heritability. Now that you're part of this class, the onus is also on you to try to explain this as well as you can, to the extent that you can. I mean, I think if there's one message that you can take home from this, it's that it's not very easy to explain, and the known genetic basis for things is rather small. Even though we have reason to believe that there's a huge influence of genetics, the amount that we know about is a very small proportion of that. That's a very simple nutshell you can put it all into. Anyway, I won't belabor this much more than I have to, because we're out of time pretty much. So I'll just get into the last slide. What to do about all of this that I've told you? Well, our study of the genetics of mental illness has left us with some tentative principles, and these are the things you can take home to your folks when you see them over the holiday, if you do, or your friends when you see them, or I mean, if you see anybody in real life, I think you're doing pretty well these days, given the whole lockdown pandemic business. But anyway, the first principle you can share is that the genetics and epigenetics of mental illness and brain phenotype are rooted in neurodevelopment for the most part. This highlights this really highlights a crucial role, I think, for early life conditions as the biggest non-genetic contributor to phenotype, meaning that if we want to make a difference, we really have to focus on early life. Second, SNP-based genetic correlations seem to suggest that there is a common architecture of mental illness that unites seemingly disparate conditions. Now, this could be because of similarity in diagnostic criteria. It might be the case that these characteristics or these traits all have similar qualities to them, and that's what's being picked up. But it also could be that there's an underlying biological architecture that we're seeing from these GWAS studies, and it's coming to us this way. And third, even the best GWAS studies we have explain a tiny fraction of inter-individual variation in illnesses and traits. This is really important to point out, is that even though we have every reason to think that some proportion of your risk of schizophrenia or depression or bipolar or whatever the case may be is genetic, we don't know for sure what those genes are. The amount of genes we have that we can really, or the amount of variance we can conclusively tie to those things is relatively small. So most of the genetic causes that we think exist are presently unknown to us and require more research to find. So our role as researchers then is to do the work responsibly, with an eye for both how the implementation and implications of this research can affect society, reinforcing hierarchy and inequality and all that kind of stuff. And if you're not a researcher, then your job is to use this knowledge to stop others from misinterpreting the data and misapplying it. After all, at this point, it's very likely that you know more about this topic than almost anybody else you'll meet, because very few people are in this exact program. Very few people you meet will have studied will have studied neuroscience and mental illness to the extent you have. Very people, only the 70 or so of you in this class have ever taken it, because it's the first time I've ever taught it, and I've never taught it anywhere else. So you're among a very small minority of people who have this exact perspective. And if you think it's valuable, your job is to at least try to apply it to your life in a helpful and meaningful way. What's next? Well, I'm done talking. I've said all I really need to say about the topic. There actually is more I could get into, but I'm trying to give you a broad survey of the field without getting too hung up in one area or another. But if you like this kind of stuff, I'll give you some suggestions from places you can follow up on and more free online content. Manolis Kallis, who I mentioned in one of the earlier lectures as a human genome or epigenome roadmap guy, has a full course on machine learning in genetics and computational biology. So if you like this kind of stuff but want to take it on in a more rigorous way, and you want to take a course that I think he's at MIT. Yeah, he's at MIT in the Broad Institute. So if you want to do like the next level version of what I'm giving you here, you can try taking this course. It's really quite good, and he's a brilliant person who I'd highly recommend listening to. If you're interested in bioinformatics from a more like how to actually do it perspective, bioinformatics.ca, which is Canadian, has an online course about the bioinformatics of genomic medicine as well. So if you want to, if you're interested in taking some time this summer or whatever to study this in more detail, these are places to look. Anyway, I've got to get on to my next meeting, my friends. So thanks to everybody for a great semester. I hope that you learned something from this. I hope that it's given you some insight into the genetics of mental health. I also thank you for your patience. This is my first time teaching this class. I've designed this material on the basis of what I think is important for you to know and what I think is interesting, but there's always a possibility that some of it hit and some of it missed. And that's, I'm aware of that. I'm trying to be understanding of that when I look at your assignments and your grades and whatnot. But hopefully at least some of it stuck with you and gave you an insight into what this area really offers. If you're excited about it, I hope you pursue it. And if you're glad to be done with the course, I hope at least something sticks with you and you learned it. So thanks to everyone for a great semester. Good luck with the exam. I'll have office hours tomorrow if you have any questions, or otherwise you can please feel free to contact me and I'll be available for you. Otherwise, good luck with the exam. Good luck with all of your other exams, and have a great break going forward. So long.